Imagine for a moment that every flower you've ever seen, every fruit you've ever tasted, every color pattern on a butterfly's wing exists because of an ancient conversation, millions of years in the making, between species that shaped each other across time. Tonight, we're going to listen in on that conversation. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. The story of how plants and animals co-evolved is not just a story of survival. It's a story of partnership, of conflict, of deception, and of dependencies so profound that some species literally cannot exist without their partners. It's a story written in nectar and pollen, in poison and antidote, in color and camouflage, in roots that reach deep into soil where fungi wait to embrace them. And it's a story that began almost as soon as life crawled onto land, more than 400 million years ago, when early plants found that they could not make this journey alone. Across the eons that followed, plants and animals have engaged in what scientists call coevolution, a process where two different organisms evolve in response to each other, each one becoming a moving target that the other must adapt to survive. When a plant evolves a new defense, herbivores evolve ways to overcome it. When animals develop new senses or abilities, plants evolve to exploit them or evade them. The result has been an explosion of diversity, an intricate web of relationships so complex that we are still discovering new threads in its weaving. Tonight, we'll walk through forests where flowers wait for specific pollinators that may visit only once a year. We'll see how a simple fruit became a sophisticated advertisement designed to catch the eye of birds flying overhead. We'll witness chemical warfare between plants and insects, fought with poisons and antidotes, refined across millions of years. We'll discover underground networks where fungi and plant roots exchange resources in partnerships older than most animal families on Earth. And we'll learn that much of the beauty we see in nature, the vibrant colors, the intricate patterns, the sweet scents, exists not for us, but as signals in conversations we can barely hear. So, settle in, let your breath slow, and prepare to see the living world not as a collection of separate species, but as a vast, interconnected dance that has been choreographed across geological time. The story of coevolution is perhaps told most beautifully in the relationship between flowers and their pollinators. About 160 million years ago, flowering plants appeared on Earth. Before this moment, plants relied primarily on wind to carry their pollen from one individual to another. A strategy that works, but requires producing enormous amounts of pollen in hopes that some small fraction might land on a receptive flower. It's inefficient, wasteful, and leaves much to chance. But then, something remarkable happened. Some plants began to recruit animals to do this work for them, and in doing so, they changed the world. The earliest pollinators were probably beetles, ancient insects that crawled across early flowers, feeding on pollen and inadvertently carrying it from plant to plant. But as flowering plants diversified and spread, so too did their animal partners. Bees, butterflies, moths, flies, birds, bats, and even some small mammals became pollinators, and plants began to evolve specific traits to attract the partners most beneficial to them. This created a feedback loop where each small change in the plant could favor a corresponding change in the animal, and vice versa, leading to increasingly specialized relationships. One of the most famous examples of this coevolutionary dance was predicted by Charles Darwin himself, long before anyone had seen it. In 1862, Darwin received a specimen of an orchid from Madagascar, a species called Angraecum sesquipedale, the star orchid. This flower possessed an extraordinary feature, a nectar spur that extended nearly a foot in length, with the sweet reward hidden at the very bottom. Darwin examined this orchid and made a bold prediction. Somewhere in Madagascar, he reasoned, there must exist a moth with a tongue long enough to reach that distant nectar. No such moth was known at the time. Critics scoffed at the idea, but Darwin was undeterred. The orchid and the moth, he argued, must have evolved together, each driving the other toward greater extremes. 41 years later, in 1903, such a moth was finally discovered. It was a hawk moth, later named Xanthopan morgani predicta, the Latin for predicted, in honor of Darwin's insight. The moth's proboscis, when fully extended, measured nearly 12 inches long, precisely matching the depth of the orchid's spur. Here was coevolution laid bare. Two species locked in an evolutionary embrace so tight that each became a mirror of the other's needs. The orchid offered nectar in a location inaccessible to most visitors, ensuring that only its specialized pollinator could reach it. The moth, in turn, gained access to a food source with little competition. Both benefited from specificity, from this narrowing of their relationship, until they fit together like pieces of a puzzle carved over millions of years. But the orchid and the moth represent just one pattern among many. Hummingbirds and the tubular flowers they pollinate show us another. Hummingbirds possess long, slender beaks and tongues that can extend even further, allowing them to reach deep into flowers. Many of the flowers they visit are red or orange, colors that stand out brilliantly to birds, but are nearly invisible to bees, which cannot see red light. These flowers also tend to be tubular, shaped to accommodate the hummingbird's beak, while keeping their nectar inaccessible to insects. They produce copious nectar to fuel the high metabolism of these tiny flying machines. And they lack strong sense, because birds, unlike insects, rely primarily on vision, rather than smell to find their food. The hummingbird's anatomy tells the other half of the story. Their rapid wing beats, their ability to hover, their long tongues. These are not random traits, but adaptations that allow them to exploit a food source that few other animals can access. The flowers benefit because hummingbirds are excellent pollinators. They visit many flowers in sequence. They fly long distances, and their movements are predictable. A hummingbird will often establish a route through its territory, visiting the same flowers day after day, ensuring reliable pollination. This consistency is valuable for plants that might be sparsely distributed across a landscape. Some relationships between plants and pollinators have become so specialized that neither partner can survive without the other. These are called obligate mutualisms, and they represent coevolution at its most extreme. Consider the fig and the fig wasp. There are hundreds of species of figs, and almost everyone has its own specific species of wasp that pollinates it. The relationship works like this. A female fig wasp, heavy with pollen from her birthplace, flies until she finds a fig tree of the correct species. When the fig is ready to be pollinated, it releases a chemical scent that attracts the wasp. She enters the fig through a tiny opening, an opening so small that she often loses her wings and parts of her antennae squeezing through. Once inside, she lays her eggs in some of the fig's flowers, pollinating others with the pollen she carries. Then she dies, her body absorbed by the growing fig. The eggs hatch, and the wasp larvae develop inside the fig, feeding on some of the seeds. When they mature, the males emerge first. These males have one purpose: to mate with the females still developing in their chambers, and then to chew an exit tunnel through the fig's wall. They never leave. They die inside the fig, having spent their entire lives in the darkness, fulfilling their evolutionary role. The females emerge, collect pollen from the male flowers that have matured inside the fig, and fly out through the tunnel the males created. They carry this pollen to another fig tree, and the cycle continues. This system is astonishingly complex and specific. The fig depends entirely on the wasp for pollination. No wasp, no seeds. The wasp depends entirely on the fig for a place to lay eggs. No fig, no next generation. This interdependence has persisted for tens of millions of years, long enough that figs and fig wasps have diversified together, each fig species maintaining its partnership with a single wasp species through all the changes the world has seen. Yucca plants and yucca moths share a similar intimacy. The female yucca moth collects pollen from yucca flowers and deliberately places it on the stigma of another flower, ensuring pollination. But before she does this, she lays her eggs inside the flower's ovary. When the eggs hatch, the larvae feed on some of the developing seeds. Not all, just some. The plant sacrifices a portion of its seeds to feed the moth's offspring, but in return, it gets guaranteed pollination. Neither the yucca nor the moth can reproduce without the other. The moth's entire life cycle is timed to match the blooming of the yucca, and the yucca's flowers only produce seeds when the moth performs her delicate act of pollination. These relationships raise a profound question. How does such specificity evolve? How does a fig tree know to release its scent at exactly the right time? How does a wasp know which fig to seek? The answer is natural selection, operating across countless generations. Any fig that released its scent too early or too late would fail to be pollinated and leave no descendants. Any wasp that couldn't recognize the right fig would waste her eggs. Over time, only the combinations that worked, the figs and wasps, whose timing and recognition were precise, would survive. Mistakes were fatal, and so the system became ever more refined. But not all plant-pollinator relationships are so exclusive. Many plants are generalists, visited by dozens of different pollinator species, and many pollinators feed from numerous plant species. These generalist relationships might seem less romantic than the obligate partnerships, but they reveal a different co-evolutionary dynamic. Instead of two species tightly bound, we see groups of plants evolving similar traits to attract groups of pollinators, creating what are called pollination syndromes. Bee-pollinated flowers, for example, tend to share certain characteristics. They are often yellow, blue, or purple, colors bees see well. They have landing platforms, structures that allow the bee to perch while feeding. They produce moderate amounts of nectar and abundant pollen. Many have ultraviolet patterns invisible to human eyes, but visible to bees. Patterns that serve as nectar guides, directing the bee to the flower's center, like runway lights guiding a plane to land. Bees have evolved to recognize these patterns and flowers have evolved to produce them. The bee's colour vision, its need for both pollen and nectar, its behaviour of collecting food to bring back to a hive, all of these traits have shaped what a bee flower looks like. And in turn, the availability of bee flowers has favoured certain bee traits, creating a feedback loop. Moth-pollinated flowers tell a different story. Many moths fly at night, so their flowers tend to be white or pale, colours that show up in moonlight or starlight. They release powerful fragrances, especially after sunset, because scent is more reliable than colour in the dark. The flowers are often tubular or have deep throats, matching the long proboscis that many moths possess. Some flowers even open only at night, timing their availability to when their pollinators are active. The moths, for their part, have evolved excellent senses of smell and the ability to hover while feeding, much like hummingbirds, but in the darkness rather than daylight. Bat-pollinated flowers are often the largest and most robust, because bats are larger than most insect pollinators and need flowers that can support their weight, or at least withstand their vigorous feeding. These flowers tend to be white or pale, visible in dim light, and they produce strong, musty odours, sometimes described as smelling like fermented fruit, or even bat colonies themselves. They open at night and produce copious nectar. Some, like certain species of agave and baobab, only bloom at night, hanging their flowers away from foliage to give bats clear flight paths. The bats, in return, provide excellent pollination services, flying long distances and visiting many plants, spreading pollen across wide areas. These patterns, these syndromes, emerged from millions of years of coevolution. Each slight advantage, a brighter colour, a stronger scent, a deeper flower tube, could mean the difference between successful reproduction and failure. And on the animal side, each improvement in sensory perception, each refinement in feeding apparatus, could mean better access to food and thus better survival. Neither plants nor pollinators were trying to evolve together. They were simply surviving, reproducing, passing on their traits. But over time, the result was partnerships that appear almost designed, so well matched that it's easy to forget they arose through blind, incremental change. When a plant produces a fruit, it is making an investment. The fruit itself is expensive to create, packed with sugars, nutrients, sometimes oils or proteins, all resources the plant could have used for other purposes. But this investment pays off because fruits are designed to be eaten. Inside each fruit are seeds, and those seeds need to travel. If they simply fall beneath the parent plant, they face intense competition for light, water, and nutrients. Far better to disperse, to find new ground where they might thrive without competing against their siblings or parent. This is where animals come in. The evolution of fruits is a story of plants learning to bribe animals. Early land plants dispersed their seeds through wind or water, methods that, like wind pollination, are scattershot and inefficient. But some plants began producing fleshy structures around their seeds, structures that animals found nutritious and desirable. When animals ate these proto-fruits, they consumed the seeds as well. And here's the key. Many seeds can survive the journey through an animal's digestive system. They pass through intact, often scarified and ready to germinate, and they are deposited far from the parent plant, conveniently packaged in a pile of fertilizer. This created a new form of coevolution. Plants began to evolve fruits specifically to attract animals that would disperse their seeds effectively. Different animals prefer different types of fruits, and plants responded by diversifying their fruit traits to match the preferences and abilities of their seed dispersers. Fruit color, size, scent, nutritional content, even the timing of ripening, all of these traits became subject to selection pressures exerted by the animals eating them. Birds became some of the most important seed dispersers, particularly for plants in tropical forests, where up to 90% of tree species produce fleshy fruits designed for birds. Birds have excellent color vision, so fruits aimed at avian dispersers tend to be brightly colored, especially in red, blue, purple, and black. These colors stand out against green foliage and signal ripeness. Birds also rely more on vision than smell, so bird-dispersed fruits often have little or no odor. They are typically small enough for a bird to swallow whole, ensuring the seeds are consumed rather than damaged. And they ripen in patterns that match the seasonal movements of migratory birds, creating a calendar of food availability that both birds and plants have come to depend upon. The fruits themselves offer different rewards. Some are high in sugars, providing quick energy for active birds. Others are rich in fats, especially valuable for birds preparing for migration or enduring cold seasons. The plant tailors its offering to the needs of its disperser, because a well-fed bird is a bird that will visit many plants and fly long distances, carrying seeds to places where they might find good germination sites. Mammals disperse seeds too, and fruits aimed at mammalian dispersers tell a different story. Mammals often have keen senses of smell, so mammal-dispersed fruits tend to produce strong odors, especially as they ripen. Think of the smell of a ripe banana, a mango, a passion fruit, scents designed to travel through the forest and alert fruit-eating mammals that food is ready. These fruits are often larger because mammals can handle bigger items. They may be yellow, brown, or green when ripe, colors that don't particularly stand out to the human eye, but that many mammals can detect through smell rather than sight. Some mammals are browsers, eating fruits where they find them and spitting out seeds nearby. Others are more mobile, carrying fruits away to eat in safe locations or hoarding them. Squirrels, for instance, are known to bury nuts and seeds, creating seed caches that they sometimes forget, inadvertently planting trees. Bears eat enormous quantities of berries during late summer and fall, and their digestive systems treat seeds gently. A bear can disperse thousands of seeds in a single day, distributing them across its large home range. The plants that bears favor have evolved fruits that ripen in late summer, exactly when bears need to put on weight for hibernation. Even fish can be seed dispersers. In flooded forests of the Amazon, fish swim among the trees during the wet season, feeding on fruits that drop into the water. Some fish have powerful jaws that can crack hard nuts, accessing the nutritious flesh inside, and then swimming away before defecating the seeds kilometers distant. These fruits are often adapted to float, making them easy for fish to find. They may also have hard, protective coatings to withstand the fish's powerful bite. The trees that produce these fruits time their fruiting to coincide with the flood season, ensuring their fruits are available when fish are swimming through the forest. The coevolution between fruits and frugivores is complicated by the fact that fruit-eating animals are generally less specialized than pollinators. A hummingbird might visit only a few species of flowers, but a fruit-eating bird might consume dozens of fruit types. This creates what scientists call diffuse coevolution, where instead of tight pairwise relationships, we see groups of plants and groups of animals evolving in response to each other's general characteristics. A plant doesn't evolve a fruit for one specific bird. It evolves a fruit that appeals to the type of bird that is common in its habitat and likely to provide good dispersal service. This diffuse coevolution can still drive remarkable adaptations. For instance, the evolution of color vision in primates may have been driven partly by the need to identify ripe fruits. Primates that could better distinguish red fruits from green leaves had a feeding advantage, and plants that could signal ripeness with color changes gained more effective dispersers. Over time, primate color vision improved, and fruit colors became more diverse and vivid. It's a feedback loop playing out across millions of years, subtle but powerful. However, not all fruit eaters are good dispersers. Some animals, like many seed predators, destroy the seeds they consume. Rodents, for example, often crack seeds to eat the nutritious interior, killing the embryo. Plants have evolved defenses against such seed predators, while still trying to attract legitimate dispersers. They might produce fruits with tough rinds that only certain animals can open, or coat seeds with chemicals that deter rodents, but don't affect birds or larger mammals, whose digestive systems can handle the compounds. The dispersal syndrome hypothesis suggests that we should be able to look at a fruit and predict what kind of animal disperses it based on its traits. A small, brightly colored berry is likely bird dispersed. A large, smelly fruit might be dispersed by mammals. A fruit that floats probably relies on water or fish. While this hypothesis holds up in many cases, reality is messier. Many fruits are eaten by multiple disperser types, and the effectiveness of dispersal can vary greatly. A fruit that evolved to be bird dispersed might also be consumed by monkeys or even bats, each animal providing different quality dispersal service. This messiness is itself a product of coevolution. When animals are abundant and diverse, plants may benefit from attracting multiple dispersers rather than relying on a single species. Generalization can be a form of insurance against the loss of any one disperser. But when dispersers are scarce or highly specialized, we see more specific fruit disperser relationships, like those between some figs and fruit bats, or certain palms and large forest mammals. The fruits we eat today, cultivated varieties of wild plants, are often the products of this ancient coevolution. Apples, grapes, berries, melons, citrus fruits, all evolved to attract animal dispersers. We have simply stepped into the role that other animals once played, selecting fruits for traits we find desirable: size, sweetness, ease of eating, and in doing so, we continue the co-evolutionary process, shaping these plants even as we consume their offerings. While pollination and seed dispersal represent co-evolution in its cooperative modes, the relationship between plants and herbivores is fundamentally antagonistic. Plants are producers, capturing solar energy through photosynthesis and storing it in their tissues. Herbivores are consumers, seeking to tap into this stored energy. But plants are not passive victims. They have evolved an arsenal of defenses to deter, poison, or otherwise discourage the animals that would eat them. And herbivores, in turn, have evolved counter-strategies to overcome these defenses. The result is an evolutionary arms race that has been running for hundreds of millions of years. Plants defend themselves with both physical and chemical weapons. Physical defenses are perhaps the most obvious. Thorns, spines, and prickles make plants difficult or painful to eat. The thorns on a rose, the spines of a cactus, the prickly surface of a thistle, these structures evolved to discourage herbivores from browsing. They're effective against larger mammals that might try to eat leaves or stems. A deer might hesitate before biting into a thorny branch. The cost of injury, the risk of infection, the pain, often outweighs the nutritional benefit. But thorns are just the beginning. Some plants have evolved hairs called trichomes that cover their leaves and stems. These hairs can be simple or branched, straight or hooked. Some are filled with irritating chemicals that break when touched, releasing compounds that sting or cause allergic reactions. Stinging nettles are a familiar example. Their leaves are covered in tiny hollow hairs that act like hypodermic needles. When an animal brushes against the plant, these hairs inject a cocktail of chemicals, including histamine, acetylcholine, and formic acid, causing intense burning pain. It's a remarkably effective defense, one that most herbivores learn to avoid. Other plants have evolved leaves that are simply tough and difficult to chew. Sclerophyll, the development of hard, leathery leaves, is common in plants growing in nutrient-poor soils. These leaves are full of lignin, the compound that makes wood woody, and cellulose fibers that resist tearing. Chewing such leaves requires strong jaws and wear-resistant teeth. Many herbivores avoid them when more tender options are available. The plants pay a cost for these tough leaves. They photosynthesize less efficiently than soft leaves. But in environments where nutrients are scarce, and losing leaves to herbivores would be costly, the trade-off makes sense. Even more sophisticated are the plants that have recruited bodyguards. Some acacias in Africa have evolved a mutualistic relationship with ants. The trees produce hollow thorns that the ants live in, and they secrete nectar from special glands that the ants feed on. In return, the ants are fiercely protective. They swarm and attack any herbivore, from tiny caterpillars to large giraffes, that tries to eat the tree's leaves. The ants even clear away competing vegetation from around the base of the tree. This is coevolution involving three parties: the plant, the ants, and the herbivores that both are defending against. But physical defenses are only half the story. Chemical defenses are often more effective and certainly more diverse. Plants produce thousands of compounds, called secondary metabolites, that have no direct role in growth or reproduction, but serve as chemical weapons against herbivores. These chemicals come in many forms: alkaloids, terpenoids, phenolics, glycosides, each with different effects on the animals that consume them. Alkaloids are nitrogen-containing compounds that can be potently toxic. Nicotine, produced by tobacco plants, is an alkaloid. It acts as a neurotoxin in insects, causing paralysis and death. Caffeine, found in coffee and tea, is another alkaloid that poisons insects and fungi, while also preventing the germination of competing plants. Morphine, cocaine, and strychnine are all plant alkaloids evolved as defenses. The fact that some of these compounds have medicinal or psychoactive effects in humans is incidental. The plants evolved them to kill or deter herbivores. Terpenoids are another large class of defensive compounds. These include the essential oils that give many plants their characteristic smells: the menthol in mint, the limonene in citrus peels, the camphor in camphor trees. Many terpenoids are volatile, evaporating easily, which is why crushing an herb or peeling an orange releases such strong scents. These compounds can be toxic to insects and fungi, disrupting cell membranes or interfering with enzyme function. Pyrethrin, a terpenoid produced by chrysanthemums, is a powerful insect neurotoxin still used in many organic insecticides today. Phenolic compounds include tannins, the same chemicals that make unripe fruit taste astringent. Tannins bind to proteins, including digestive enzymes, making food harder to digest. A herbivore that eats leaves full of tannins will gain less nutrition from its meal, even if it can tolerate the bitter taste. Over time, this reduced nutritional payoff can lead the herbivore to seek other food sources. Some of the most toxic plant defenses are glycosides, compounds that release poisons when plant tissues are damaged. Cyanogenic glycosides, found in plants like cassava and certain wild cherries, release cyanide when the plant is chewed. Cardiac glycosides, found in plants like foxgloves and milkweed, interfere with the sodium-potassium pumps in animal cells, causing heart arrhythmias and death at high doses. This chemical warfare is astonishingly effective, but herbivores have not remained defenseless. They have evolved counter-adaptations, ways to tolerate, neutralize, or even exploit plant toxins. This is where coevolution becomes most dramatic. A back-and-forth escalation where each new plant defense selects for herbivores that can overcome it, and each new herbivore adaptation selects for even stronger plant defenses. Some herbivores have evolved detoxification mechanisms. Their digestive systems produce enzymes that break down plant toxins, rendering them harmless. The liver is a key organ in this process, filtering blood and metabolizing toxins before they can reach critical organs. Herbivores that feed on toxic plants often have enlarged livers and high levels of detoxification enzymes. This is energetically expensive, another cost in the evolutionary arms race. But it allows access to food sources that other animals can't use. Other herbivores have become specialists, feeding on only one or a few plant species. These specialists often evolve specific adaptations to handle the particular toxins their host plants produce. The tobacco hornworm caterpillar, for instance, feeds exclusively on tobacco and related plants. It can tolerate nicotine levels that would kill most other insects. The caterpillar's cells have modified nicotine receptors that are resistant to the toxin's effects, and it can excrete excess nicotine rapidly. Perhaps most remarkable are the herbivores that have learned to sequester plant toxins, storing them in their own bodies to use for defense. Monarch butterflies are the most famous example. As caterpillars, they feed on milkweed plants, which contain cardiac glycosides. The caterpillars not only tolerate these toxins, but actively store them in their tissues. When they metamorphose into adult butterflies, they retain the toxins, making themselves poisonous to predators. A bird that eats a monarch butterfly will become violently ill, and it will remember the experience. The butterfly's bright orange and black coloring serves as a warning, a signal that says, "I am poisonous, do not eat me." This warning coloration, called aposematism, only works because the butterfly has hijacked the milkweed's chemical defenses and repurposed them. This sets up a fascinating co-evolutionary dynamic. The milkweed evolves toxins to deter herbivores. The monarch evolves to tolerate and sequester those toxins. But some milkweeds have responded by evolving even more potent toxins, or different forms of cardiac glycosides that are harder for the butterfly to handle. The monarch, in turn, continues to adapt. We can see this evolutionary arms race playing out in real time by looking at different populations. Monarchs that feed on more toxic milkweed species have evolved higher resistance to cardiac glycosides than monarchs feeding on less toxic species. The plants and the butterflies are still evolving, still engaged in their ancient dance. A similar story plays out between wild parsnips and parsnip webworms. Wild parsnips produce compounds called furanocoumarins, chemicals that bond with DNA when exposed to UV light, causing severe cellular damage. These compounds are derived from simpler molecules found throughout the carrot family. But furanocoumarins represent an evolutionary innovation, a new weapon in the plant's arsenal. Most herbivores cannot tolerate furanocoumarins, but the parsnip webworm can. It has evolved enzymes that detoxify these compounds, and it feeds on wild parsnip with impunity. But the story doesn't end there. Researchers have found that wild parsnip populations vary in the types and concentrations of furanocoumarins they produce. Some plants produce more of one type, others more of another. And the webworms show corresponding variation in their ability to detoxify different furanocoumarin types. Where the plants produce more of one compound, the local webworm population has evolved stronger detoxification of that specific compound. This is coevolution on a fine scale. Different populations of plants and herbivores engaged in their own localized arms races, each adapting to the specific challenges posed by their local partners. Some plants take a different approach. Instead of producing toxins continuously, which is expensive, they produce them only when attacked. These induced defenses allow the plant to save resources until they're needed. When a plant is damaged, chemicals in the herbivore's saliva or the plant's own damaged cells trigger a cascade of defensive responses. Within hours or days, the plant can increase the concentration of toxins in its leaves, produce new defensive compounds, or even change the structure of its leaves to make them tougher. Some plants also produce volatile organic compounds when damaged, scents that travel through the air and serve multiple functions. These VOCs can attract predators or parasites of the herbivores, recruiting help in defending the plant. For example, when a caterpillar eats a cabbage plant, the plant releases chemical signals that attract parasitic wasps. These wasps lay their eggs inside the caterpillar, and when the eggs hatch, the wasp larvae eat the caterpillar from the inside out. The plant doesn't just defend itself, it calls in an ally, and the wasps have evolved to recognize and respond to these chemical cues because they signal the presence of their own prey. The arms race between plants and herbivores has driven much of the diversification we see in the insect world. There are more species of herbivorous insects than any other group of animals, and this explosion of diversity is thought to be linked to the evolution of new plant defenses and the herbivores that overcame them. When a lineage of insects evolves the ability to feed on a previously inaccessible group of plants, it opens up new ecological opportunities. Those insects can radiate, diversifying into many new species, each adapted to different plants in that group. This process, sometimes called escape and radiation, has happened repeatedly throughout evolutionary history. The defenses we see in plants today, the thorns and toxins, the toughness and the calls for help, are the accumulated results of millions of years of this co-evolutionary struggle. And herbivores carry their own history, written in their detoxification enzymes, their specialized digestive systems, their behaviors that minimize toxin exposure. Neither side is winning. Both continue to evolve, locked in a dance that has no endpoint, only the constant pressure of selection, pushing each toward adaptations that might provide, if only temporarily, an edge in the ongoing contest. Not all coevolution involves direct chemical warfare or nutrient exchange. Sometimes it involves deception, disguise, and mimicry, strategies that play out through visual signals and the eyes that perceive them. In the evolutionary theatre of colour and pattern, plants and animals have evolved some of nature's most extraordinary masquerades. Camouflage, the art of blending into one's surroundings, is a form of coevolution between prey and predator. When a prey animal evolves better camouflage, predators face a harder task in finding food. This selects for predators with sharper vision, better search strategies, or alternative hunting methods. As predators improve, the pressure on prey to hide more effectively intensifies. The result is an arms race of concealment and detection that has produced some of the most remarkable adaptations in nature. The walking stick insect is a master of disguise. Its body is elongated and twig-like, coloured in browns and greens that match the branches where it rests. Its legs are held in positions that mimic twigs extending from the main branch. It even moves in a swaying motion, mimicking the movement of vegetation in a breeze. To a predator scanning the foliage for a meal, the walking stick is nearly invisible. This disguise evolved because any ancestral insect that happened to be slightly more stick-like than its siblings had a better chance of surviving to reproduce. Over countless generations, natural selection refined the disguise until it became nearly perfect. But predators have not stood still. Birds that hunt insects have evolved excellent pattern recognition. They learn to identify the subtle cues that give away a camouflaged insect, a shadow that doesn't quite match, a texture that's slightly off, a movement that's too deliberate. This forces prey to evolve even better camouflage. Some walking stick species have gone beyond mere resemblance to sticks. They have evolved bumps and ridges that mimic leaf scars or patches of lichen-like colouring. They may have body segments that look like broken or decaying wood. The deception has become multi-layered, anticipating the predator's ability to see through simpler disguises. Other insects mimic leaves, dead or alive. The dead leaf butterfly, when at rest with wings folded, looks almost exactly like a dried brown leaf, complete with what appears to be a midrib, and even spots that resemble patches of decay or insect damage. Only when it flies does the deception end, revealing the brightly coloured upper surfaces of its wings. But by then, it may have evaded a predator's initial strike. Camouflage also involves behaviour. The insect must rest in the right location, on the right background, and remain still when predators are near. This behavioural component means that camouflage is not just about looking right, but about acting right. And it means that predators must evolve, not just to see through the disguise, but to recognise the behavioural patterns that might betray hidden prey. Some prey species take the opposite approach. Instead of hiding, they advertise. This is warning coloration, or aposematism, and it works only if the advertiser has something to back up the warning. Poisonous frogs in tropical rainforests are often brilliantly coloured, red, yellow, blue, orange, in combinations that stand out starkly against the green of the forest floor. These colours are a signal to predators. I am toxic. Eating me will make you sick or kill you. Don't try. Warning coloration only makes sense if predators can learn. A poison dart frog doesn't benefit from being bright if a predator eats it and dies. The frog is still dead. But if the predator survives the encounter, even if it becomes ill, it can learn to associate those colors with danger and avoid similar frogs in the future. This requires that predators have memory and the capacity to associate visual cues with past experiences. Birds, mammals, and even some reptiles possess these cognitive abilities, and this has allowed warning coloration to evolve. The coevolution here is subtle. The prey evolves bright colors and toxicity. The predator evolves the ability to recognize and remember those colors. Young predators that make mistakes, eating toxic prey, suffer the consequences but learn. Over generations, predators that are quicker to learn, that have innate biases toward avoiding certain color patterns, or that can generalize from one bad experience to avoid similar looking prey in the future, will be more successful. Meanwhile, prey species that can produce more reliable, more conspicuous signals will be better protected. This sets the stage for mimicry. If bright colors protect toxic prey from predators, then a non-toxic species could benefit by resembling a toxic one. This is Batesian mimicry, named after the naturalist Henry Walter Bates, who first described it in the 1800s after studying butterflies in the Amazon. The viceroy butterfly, which is harmless, closely resembles the monarch butterfly, which is toxic from the milkweed it consumed as a caterpillar. Birds that have learned to avoid monarchs also avoid viceroys, even though eating a viceroy would cause no harm. Batesian mimicry is evolutionary deception. The mimic gains protection without paying the cost of producing toxins. But for this to work, the mimic must be less common than the model. If predators encounter more viceroys than monarchs, they will learn that butterflies with this color pattern are usually safe to eat, and the protection breaks down. So viceroys are typically less abundant than monarchs in areas where they coexist. The ratio is maintained by the fact that if viceroys become too common, predators start eating them, and their population declines until the balance is restored. There is another form of mimicry where both species are toxic, called Müllerian mimicry, after the naturalist Fritz Müller. In this case, multiple toxic species evolve to resemble each other. Each species benefits because predators learn to avoid the shared color pattern more quickly. If a bird tries one toxic species and becomes ill, it will avoid all species that look similar. This spreads the cost of educating predators across multiple species. No single species has to sacrifice as many individuals to teach predators the lesson. Bees and wasps, many of which sting, often display Müllerian mimicry. They share similar black and yellow banding patterns, a warning that says we sting. A bird that gets stung by a wasp will avoid bees, and vice versa. The similarity in coloration benefits both because it reinforces the warning. This convergent evolution of color patterns creates what are called mimicry rings, groups of species that all resemble each other and all share some form of defense. But mimicry isn't limited to warning colors. Some prey mimic inanimate objects to avoid detection. The aforementioned deadleaf butterfly is an example. So are stick insects and leaf insects. Some caterpillars mimic bird droppings, looking like a white and brown blob on a leaf, something a predator would ignore. Others mimic twigs or broken branches, complete with realistic texture and coloring. Predators themselves sometimes use mimicry. The alligator snapping turtle has a tongue that looks remarkably like a small worm or piece of meat. It lies on the bottom of a river with its mouth open, wiggling this lure. Fish, attracted by what appears to be an easy meal, swim close to investigate. The turtle's jaws snap shut with crushing force. The lure evolved through coevolution with the turtle's prey. Fish that are attracted to worm-like movements have selected for turtles with more convincing lures, and the most curious fish have been removed from the population, leaving more wary individuals. The coevolutionary feedback continues, predator and prey each shaping the other's evolution. Even plants engage in mimicry, though their targets are usually pollinators rather than predators. Some orchids mimic the appearance and even the scent of female insects. Male insects, fooled by the deception, attempt to mate with the flower, and in doing so, pick up pollen. The insect gets nothing from the interaction, no nectar, no mate, just a wasted effort. But the orchid is pollinated. This form of mimicry, called sexual deception, has evolved independently in several orchid lineages, always in response to the sensory biases and mating behaviors of the insects they mimic. The evolution of such complex mimicry requires that small changes in appearance or scent, even if initially imperfect, provide some benefit. Perhaps an orchid that vaguely resembled an insect attracted slightly more insect visitors than others. Over time, natural selection would favor those orchids that resembled the insect more closely, refining the deception. Meanwhile, the insects might evolve to be more discriminating, to better distinguish between real mates and orchid mimics. But if the cost of occasionally being fooled is low compared to the cost of missing a real mating opportunity, the insects may remain vulnerable to the deception. The orchid and the insect are locked in a co-evolutionary relationship, though one far more one-sided than most mutualisms. Beneath the soil, hidden from view, one of the most ancient and consequential partnerships on Earth plays out. It is a partnership between plant roots and fungi, a relationship so old that it may have been essential to the very colonization of land by plants more than 400 million years ago. This is the mycorrhizal symbiosis, and it represents coevolution at its most fundamental. The word mycorrhiza means fungus root, and it describes an intimate association where fungal threads, called hyphae, intertwine with or penetrate plant roots. The fungus provides the plant with nutrients, particularly phosphorus and nitrogen, which it extracts from the soil with far greater efficiency than roots alone could achieve. In return, the plant provides the fungus with sugars produced through photosynthesis. Both partners benefit. Neither could thrive as well alone. The origins of this partnership reach back to a time when the earliest land plants were small, simple organisms lacking true roots. These pioneer plants faced enormous challenges. Soil, as we know it today, barely existed. What passed for soil was little more than weathered rock and decaying microbial mats. Extracting nutrients from this substrate was difficult, but fungi were already present, living in the damp spaces between mineral particles, feeding on organic matter and breaking down rock through the acids they secreted. Somewhere in this ancient landscape, a partnership formed. A fungus colonized a plant, perhaps initially as a pathogen, or simply as an opportunist seeking the sugars the plant produced. But instead of harming the plant, the fungus provided benefits. Its hyphae, far finer than any root hair, penetrated tiny spaces in the substrate, accessing water and nutrients the plant couldn't reach. The plant, in turn, provided the fungus with a reliable source of carbon. This was the beginning of mycorrhizae, and it may have been one of the key innovations that allowed plants to spread across the land. Today, more than 80% of plant species form mycorrhizal associations. There are several types, but the most ancient and widespread is arbuscular mycorrhiza, where the fungal hyphae actually penetrate into the cells of the plant root, forming tiny, tree-like structures called arbuscules. These arbuscules are the sites of nutrient exchange. The fungus delivers phosphorus, nitrogen, and other minerals to the plant, and the plant delivers sugars to the fungus. The exchange is highly regulated, mediated by cellular machinery that both partners have evolved to control. If one partner tries to cheat, taking without giving, the other can respond by shutting down the exchange. This reciprocal exchange is a form of biological market. The plant and the fungus are trading, and like any market, it only works if both parties benefit. But this raises questions. How did such a finely tuned system evolve? How do the partners recognize each other? How do they coordinate their exchanges? The answers lie in coevolution. Plants and mycorrhizal fungi have been associated for so long that they have evolved molecular signals to communicate with each other. When a plant root grows, it secretes chemical compounds into the soil. Mycorrhizal fungi detect these signals and respond by growing toward the root. The fungus, in turn, releases its own signals, molecules that the plant recognizes. These signals trigger changes in the root, preparing it for colonization. Cells rearrange, membranes become more permeable, and the plant begins to allocate resources towards supporting the fungal partner. Once the partnership is established, the fungal hyphae spread through the soil, creating a network that can extend for meters beyond the root. This network vastly increases the surface area for nutrient absorption. A single plant connected to a mycorrhizal network can access nutrients from a volume of soil hundreds of times larger than it could with roots alone. Phosphorus, which is often limiting in many ecosystems because it doesn't move easily through soil, becomes accessible. The fungus brings it directly to the plant, but the partnership goes deeper. Recent research has revealed that mycorrhizal fungi can connect multiple plants, forming underground networks, sometimes called the Wood Wide Web. Through these networks, resources can move from one plant to another. A large tree connected to the network might share sugars with a seedling struggling in its shade. Nutrients can flow from a dying plant to healthy neighbors. Information, in the form of chemical signals, might even be transmitted. If one plant is attacked by insects, it might send chemical warnings through the mycorrhizal network, prompting neighboring plants to activate their own defenses. The existence of these networks raises profound questions about individuality and cooperation in plants. Are the trees in a forest separate individuals competing for resources? Or are they part of a superorganism, interconnected and mutually supportive? The answer is probably somewhere in between. Competition and cooperation coexist. The mycorrhizal network allows for sharing, but plants still compete for light, space, and nutrients. The network itself is not altruistic. It exists because both plants and fungi benefit from the connections. Another crucial partnership involving microbes and plants is nitrogen fixation. Nitrogen is abundant in the atmosphere, making up 78% of the air we breathe. But in this gaseous form, it is unusable by most organisms. To be useful, nitrogen must be fixed, converted into forms like ammonia that can be incorporated into organic molecules. Only certain bacteria and archaea have evolved the ability to perform this conversion, a metabolically expensive process that requires specialized enzymes. Some of these nitrogen-fixing bacteria have entered into partnerships with plants, particularly legumes like beans, clover, and peas. The bacteria live in special structures called root nodules that the plant forms in response to bacterial signals. Inside these nodules, the bacteria fix nitrogen from the air and share it with the plant. In return, the plant provides the bacteria with sugars and protected, oxygen-controlled environment. The process of nitrogen fixation is inhibited by oxygen, so the nodules maintain low oxygen levels, allowing the bacteria to do their work. This partnership, like mycorrhizae, is the product of millions of years of co-evolution. The plants have evolved the ability to recognize specific bacterial species, to form nodules in response to their signals, and to regulate the partnership. The bacteria have evolved to live inside plant tissues, to respond to plant signals, and to share the nitrogen they fix. Both have evolved molecular machinery to communicate and coordinate their activities. Interestingly, the genes involved in forming mycorrhizal associations and root nodules overlap significantly. This suggests that the nitrogen-fixing symbiosis may have evolved from the mycorrhizal symbiosis, co-opting existing pathways for a new purpose. Evolution often works by repurposing existing structures and systems, and the similarities between these two partnerships hint at a shared evolutionary origin. The benefits of these microbial partnerships are enormous. A plant with mycorrhizae can grow in nutrient-poor soils where unassociated plants would struggle. A legume with nitrogen-fixing bacteria can thrive in nitrogen-depleted environments. These partnerships have allowed plants to colonize habitats that would otherwise be inhospitable, and they play critical roles in ecosystems around the world. In forests, mycorrhizae connect trees to the nutrient cycle, recycling minerals from decaying organic matter. In grasslands and agricultural fields, nitrogen-fixing plants enrich the soil, providing nitrogen that other plants can use after the legume dies and decomposes. But these partnerships are not without costs. The plant must provide substantial amounts of sugar to its microbial partners. Sugar that could have been used for growth or reproduction. The partnerships only make sense when the benefits, improved nutrition, outweigh the costs, and this balance can shift depending on environmental conditions. In nutrient-rich soils, mycorrhizae provide less benefit, and some plants may reduce their investment in the partnership or form fewer associations. In very poor soils, the partnership becomes essential. This flexibility is itself a product of co-evolution. Plants that can adjust their investment in mycorrhizae based on nutrient availability will allocate resources more efficiently than plants that maintain the partnership regardless of need. The fungi, too, benefit from being able to colonize plants selectively, investing more in partnerships that provide better returns. The result is a dynamic relationship, one that can shift from mutualism to parasitism, depending on circumstances, but that, on average, benefits both partners enough to have persisted across geological time. Coevolution is not just about matching traits or trading resources. It also involves time, the synchronization of life cycles, the timing of events so that partners meet when they need each other most. This temporal dimension of coevolution has shaped some of the most remarkable patterns in nature. Consider the relationship between flowering plants and their pollinators. A flower that blooms when no pollinators are active will fail to reproduce. A pollinator that emerges when no flowers are available will starve. Natural selection has synchronized the timing of flowering and pollinator activity so that they match. In temperate regions, many plants bloom in spring when bees and other insects emerge from winter dormancy. The exact timing can be critical. Plants that bloom a week too early or too late may miss the peak of pollinator activity and produce fewer seeds. This synchronization is so precise that climate change is now disrupting it in some systems. As temperatures warm, spring arrives earlier, and many plants are flowering sooner. But if their pollinators are responding to different cues, perhaps day length rather than temperature, they may not emerge earlier. The mismatch can leave plants unpollinated and pollinators without food. This is a coevolutionary relationship being pulled apart by environmental change, a reminder that these partnerships are not fixed but depend on stable environmental conditions. Migratory birds and the plants that feed them show another form of temporal coevolution. Many trees in tropical forests time their fruiting to coincide with the arrival of migratory birds. The birds, traveling thousands of kilometers, arrive hungry and eager for the fruit. The trees benefit from having mobile, long-distance seed dispersers, available exactly when their fruits ripen. If a tree fruited too early or too late, it might miss the migrants and rely instead on resident birds, which may not disperse seeds as far or to as many different locations. Some plants have taken this synchronization to extremes. Bamboo species are famous for their synchronized flowering. Individual bamboo plants can grow for decades without flowering. Then suddenly, all the plants in a population flower at once, produce seeds, and die. The entire population is synchronized, sometimes across hundreds or thousands of square kilometers. The selective advantage of this synchronization is thought to be predator satiation. Bamboo seeds are eaten by rodents. By producing enormous numbers of seeds all at once, the bamboo overwhelms the rodents. They can't possibly eat all the seeds, so many survive to germinate. If individual plants flowered at different times, rodents could focus on each flowering event and consume most of the seeds. This mass flowering is a coevolutionary strategy against seed predators. The rodents, unable to eliminate the seed crop, have not evolved a counter-strategy. They simply gorge during the bonanza years, and their populations explode. But by the time the rodent population has grown large enough to potentially consume all the seeds, the bamboo has finished flowering and won't flower again for decades. The rodent population crashes from lack of food, and the cycle resets. The bamboo and the rodents are engaged in a coevolutionary dance across timescales of decades. A dance that neither fully controls, but both are shaped by. Periodical cicadas show a similar pattern. These insects spend 13 or 17 years underground as nymphs, then emerge all at once in massive numbers, mate, lay eggs, and die. The synchronization is thought to have evolved as a defense against predators. Birds, mammals, and other predators that eat cicadas can gorge during emergence years, but they can't eat them all. The sheer number of cicadas ensures that many survive. The prime number life cycles, 13 or 17 years, are thought to make it difficult for predators with shorter life cycles to synchronize with the cicadas. A predator with a 2-year or 3-year cycle would only occasionally coincide with the cicada emergence, not often enough to specialize on them. Other examples of temporal coevolution are more subtle but no less important. Many plants produce fruits that ripen sequentially rather than all at once. This staggered ripening ensures that frugivorous animals keep visiting the plant over an extended period, increasing the chances of seed dispersal. But it also means that the plant must balance the risk of some fruits being eaten by non-dispersers or rotting before they are eaten against the benefit of attracting dispersers over a longer time window. Some fruiting plants have evolved to ripen their fruits in a gradient from the bottom of the plant to the top, or from the outer branches to the inner branches. This pattern can match the foraging behavior of dispersers, which might move through the canopy in predictable ways. A bird that feeds low in the canopy first might preferentially disperse seeds from fruits that ripen low first. The plant's fruiting pattern has evolved to match the disperser's foraging pattern, a form of temporal and spatial synchronization. Even predator-prey relationships involve temporal coevolution. Many prey species have evolved to be active at times when their predators are not. Nocturnal animals avoid diurnal predators. Diurnal animals avoid nocturnal predators, but predators can adapt. Some owls hunt primarily at dawn and dusk, when the transition between day and night creates opportunities to catch both diurnal and nocturnal prey. Prey species must then adjust their activity patterns to avoid these crepuscular periods, and the back and forth continues. The timing of reproduction in many animals is also shaped by coevolution with the plants or animals they eat. Herbivorous insects often time their egg-laying to coincide with the emergence of new plant growth, when leaves are tender and nutritious. The plants, in turn, may vary the timing of their growth to avoid peak herbivore activity, or they may produce defensive compounds specifically during the periods when herbivores are most active. This creates a temporal arms race, each side trying to shift its timing to gain an advantage. These temporal patterns are not fixed. They can evolve relatively quickly in response to changing selection pressures. This means that as climates change, as invasive species arrive, as ecosystems are disrupted, the temporal synchronies that coevolution has produced over millennia can be thrown into disarray. The consequences can ripple through entire ecosystems, affecting species that were never directly involved in the original coevolutionary relationship. Coevolution is not a relic of the past. It continues today, shaping species in real time, often in response to human activities. We are witnessing evolutionary change at speeds that would have seemed impossible just a few decades ago, driven by the pressures we impose on the natural world. One of the most striking examples comes from agriculture. The use of pesticides has created intense selection pressure on insect populations. Insects that happen to have slight resistance to a pesticide survive when others die. They reproduce, passing on their resistance genes. Within a few generations, entire populations can become resistant. Farmers respond by using stronger pesticides or different chemicals. The insects adapt again. This is an arms race playing out in fields around the world, driven by human intervention, but following the same coevolutionary dynamics that have shaped plants and herbivores for millions of years. The same process occurs with herbicides and weeds. Plants that happen to be resistant to a particular herbicide survive and spread. Some weeds have evolved resistance to multiple herbicides, becoming superweeds that are difficult or impossible to control with chemicals alone. Farmers are forced to return to mechanical weeding or to develop new herbicides, continuing the cycle. The speed of this evolution, sometimes just a few years from the introduction of a herbicide to the appearance of resistance, demonstrates that coevolution is not a slow, gradual process. When selection pressure is strong, evolution can be rapid. Antibiotic resistance in bacteria is another example, though here the coevolution is between microbes and the drugs we use to kill them. Bacteria evolve resistance to antibiotics through mutations or by acquiring resistance genes from other bacteria. Doctors respond by prescribing different antibiotics. The bacteria adapt again. This coevolutionary race has profound implications for human health, as we are running out of effective antibiotics for some infections. The bacteria, with their short generation times and enormous population sizes, can evolve far faster than we can develop new drugs. Climate change is altering coevolutionary relationships in less direct but equally significant ways. As temperatures rise and seasons shift, the timing of flowering, pollinator emergence, migration, and other critical events is changing. When these changes happen at different rates, co-evolved partnerships can fall out of sync. A plant might bloom before its pollinators arrive, or a migratory bird might reach its breeding grounds after the peak abundance of the insects it feeds. Its chicks has passed. These mismatches can reduce reproductive success for both partners, and if they persist, natural selection will favor individuals that can adjust their timing. Evolution will begin to resynchronize the relationship, but this takes time, and in the meantime, populations may decline or ecosystems may shift to new configurations. Invasive species create novel coevolutionary dynamics. When a plant or animal is introduced to a new region, it encounters species it has never evolved with. The lack of coevolutionary history can be advantageous for the invader. An invasive plant may encounter herbivores that haven't evolved to eat it, and thus have no resistance to its defenses. Without herbivores to control it, the plant can spread unchecked. Conversely, an invasive herbivore might find native plants that lack defenses against it. But over time, coevolution can begin. Native herbivores may start to feed on the invasive plant, evolving tolerance to its novel toxins. Native plants may evolve new defenses against the invasive herbivore. The ecosystem begins to adapt, though the process can take decades or centuries, and in the meantime, the invader may have caused significant disruption. Some researchers are even exploring the possibility of using evolutionary principles to manage ecosystems. By understanding coevolutionary relationships, we might design conservation strategies that preserve not just species, but the interactions between them. Protecting a rare orchid, for instance, requires protecting its pollinator. Maintaining a fruiting tree in a forest requires ensuring that the animals that disperse its seeds are present. Ecosystem management is increasingly recognizing that species do not exist in isolation, but are embedded in webs of co-evolved relationships. The study of coevolution also informs our understanding of how ecosystems might respond to future changes. If we know which relationships are tightly co-evolved, we can predict which might be most vulnerable to disruption. A highly specialized pollinator and its host plant might both be at risk if climate change desynchronizes their life cycles. A generalist pollinator that visits many plant species might be more resilient. By identifying the keystones in the network of co-evolved relationships, we can focus conservation efforts where they will have the greatest impact. Looking forward, coevolution will continue to shape the living world. As long as organisms interact, as long as some benefit from those interactions and others are harmed, as long as traits can be inherited and selection can act, coevolution will proceed. The arms races, the mutualisms, the deceptions, all will continue, though perhaps in new forms, responding to new pressures in a world increasingly dominated by human influence. One of the most profound realizations from studying coevolution is that no species evolves alone. We are all products of interactions, our traits shaped by the other organisms we encounter. The flower exists because of the pollinator. The pollinator exists because of the flower. The toxic plant and the herbivore that can tolerate its toxins are bound together in a dance that neither leads. The mycorrhizal network connects trees underground, allowing them to share resources, so that a forest is not merely a collection of individuals, but a community linked by invisible threads of evolutionary history. This interconnectedness means that when we change one species, we change its partners as well. When we drive a pollinator to extinction, the plants it pollinated may follow. When we alter a plant's habitat, the animals that depend on it must adapt or disappear. The web of co-evolved relationships is resilient in some ways, capable of absorbing shocks and reorganizing, but it is also fragile. Break enough threads, and the web may collapse. The story of coevolution is ultimately a story about relationships. It reminds us that life is not solitary, that survival depends not only on individual fitness, but on the fitness of the partnerships we form. In a world that often emphasizes competition and conflict, coevolution shows us that cooperation, mutualism, and interdependence are equally powerful forces shaping the living world. The beauty of a flower, the sweetness of a fruit, the warning flash of a butterfly's wing, these are not arbitrary. They are signals in conversations that have been ongoing for millions of years. Conversations between species learning to live with and through each other. As we draw tonight's exploration to a close, consider this. The next time you see a bee visiting a flower, or eat a piece of fruit, or notice a caterpillar on a leaf, you are witnessing just a brief moment in a relationship that stretches back across deep time. The bee and the flower, the fruit and the bird, the caterpillar and the plant, they are all part of an ancient dialogue, a co-evolutionary conversation still unfolding. We, too, are part of this conversation. The plants we cultivate, the animals we domesticate, even the microbes in our bodies, all have co-evolved with us to some degree. We are not separate from nature's web. We are woven into it. And that, perhaps, is the most profound lesson of all. In understanding co-evolution, we see not just how species shape each other, but how everything is connected, how the past shapes the present, and how the choices we make today will ripple through evolutionary time, shaping the partnerships of tomorrow. Thank you for spending this time with me tonight. If you found this journey through the co-evolved relationships of the natural world helpful for relaxation, or if it sparked your curiosity, please consider liking this video and subscribing to Bedtime Biology. Your support helps us continue creating these calm explorations of science. And if you have suggestions for future topics, I'd love to hear them in the comments below. Until next time, sleep well and may your dreams be filled with the quiet wonders of the living world.